let's take a look at what Lab 4A results should look like, could look like, and what may affect these could look like cases. A reminder again, here is the full P-ARR plasmid that we have digested with restriction enzymes in Lab 2A, with the BAMH1 and HindE3 restriction sites shown on the plasmid map. And these are the two fragments that should be in the R+ sample if the restriction digest was successful, an 807 base pair fragment and a 4,495 base pair fragment. The Lab 4 setup seen here is from the ABE Canada kit, using the mini PCR blue gel system. Keep in mind that your site may offer different versions of any or all of the equipment shown here. This is an accelerated time-lapse video of the R+ and R- samples run through a 0.8% agarose gel, courtesy of students and teachers from ABE San Francisco. The marker is in the bottom lane, with R- in the middle lane and R+ in the top lane. You can see the fragments separating quite clearly in the R+ lane, and the different plasmid configurations just starting to separate in the R- lane. This image depicts a pretty good set of Lab 4A results, with the marker on the left, R- in the middle, and R+ on the right. The DNA marker is also called the DNA ladder, and we use them interchangeably. It's called a ladder because of its physical appearance, with the DNA bands resembling rungs on a ladder. On the right side here, I've zoomed in on the top section of the gel to show the bands in the R- lane. The top section of multiple bands that look like a large smear are likely multimeric plasmids, doubles or triples or multiples that are in the relaxed circular form. The distinct DNA band just below that should be the single relaxed circular plasmid. And finally, the bottom band should be the supercoiled plasmid. For a refresher on supercoiled DNA, please refer to the plasmid configuration session and the resources within that section. In the R+ lane, we see the restriction digested samples of the P-ARR plasmid, with the 807 base pair and 4,495 base pair fragments running where they would be expected to run on the gel. There is little DNA around the supercoiled or circular DNA positions, which indicate that the digest was likely fully completed. This is, of course, an ideal gel, except for the small waviness in the marker lane that could be an indication of a small tear in the well when the samples were loaded into the gel. Now, of course, not everything always goes according to plan, as illustrated by some of these following images courtesy of the ABE Los Angeles team. This first gel isn't too bad, as you see nice clean bands in the R+ and R- lanes. However, the marker lane well and possibly the R+ lane well likely had some small tears to make that wavy shape in the DNA band as you saw in the previous gel. You also see a bit of a shadow that corresponds to the uncut DNA plasmid location in the R+ lane, so the restriction digest probably was not fully completed. In the second gel, there is clearly a tear or a puncture in the R+ well that could have occurred during casting, if the comb was removed incorrectly, for example, or something happened during the sample loading process. As a result, the gel ran quite poorly around that region. Given the shape of the smear around that area, there may have been a finger used to salvage the tear in the well, resulting in the fingerprint-shaped smear in the gel itself. The gel was also not run long enough to see a full separation of the R- plasmid configuration. This third gel has a wavy pattern or a W pattern in the marker, indicating that the gel was likely run with either old running buffer or overheated running buffer, as well as what appears to be quite overloaded R- and R+ lanes. This is sometimes seen when running samples through thinner gels or when more than one sample is loaded into the same well by mistake. Now in this last gel, the marker ran quite well, indicating that the gel was likely made and ran correctly. However, we're not entirely sure where that R+ sample went. Maybe it wasn't loaded properly, or maybe it was a bubble in the well that pushed out the sample. In addition, the R- lane also does appear to be quite overloaded. You should be prepared to assist your students in making sense of their results, which may differ from the expected results, especially in this lab where so many things could go wrong. Remember, unexpected results are often when scientific breakthroughs are made.